Today what I want to do is I want to finish up with a couple of slides and tell two fairly simple stories, one of which is this idea of the cytoskeleton and long distance and short distance transport. And that all started yesterday when we were laying down the components of the rails along which we moved molecules using those molecular motors. Now, we talked about microtubules, we talked about the motors on those being kinesin and dynein, we talked about actin, filamentous actin, with its motor, the myosin. And what I want to finish with is a sort of general story, and there's some controversy to the story yet, but a general story about long distance and short distance deliveries, which will, to some extent, be akin to bringing a FedEx package to your house first by rail and then by local truck. And I'll talk to you a little bit about that. But most of today's discussion is going to be about the central dogma. The central dogma is a fundamental idea in biology, and we've mentioned it in passing before. And basically, we're now going to drill down into why DNA and how DNA is the information storage molecule and why this means genes. And then from there, we'll then look at the relationship between DNA and RNA. This is a nucleic acid as well as RNA, but there's subtle differences between the two. And the RNA, particularly in the form of messenger RNA, is going to be an intermediate between DNA and RNA. And then finally, the information that was stored in the genes up here in the DNA is now going to be expressed as an amino acid sequence in protein. So we'll see how all of that gets done. We understand this in exquisite detail. And now, in our modern world, we can co-opt this to doing our own bioengineering, for better or for worse. And we'll discuss that a little bit. But to really understand how any of this works, to understand the fundamentals of the central dogma, we pretty much have to begin with DNA structure. And it's important to recognize as we walk through this, the philosophical view, that DNA is the building block of life on the planet as we know it. It obeys the rules of the fundamental molecule of life. That is, first, it has to be able to replicate itself. One of the characteristics of all life is it makes new copies of itself. Well, this molecule fits that bill. It makes new copies of itself. But the other characteristic of this molecule, which really tells us that it's the heart of life on our planet, is that in addition to replicating itself, it can change. This is each time it makes a replicate of itself, there's a possibility it may make a mistake, a small possibility. And for the most part, those mistakes are detrimental. But now and again, the mistake is made, which leads to a slightly different gene, which leads to a slightly different protein, which leads to a slightly different benefit for the animal to survive. And so we call this evolution. And evolution is really, in many other ways, there's evolution, but DNA lies at the heart of it. And DNA lies at the heart of it because it's the molecule responsible for being able to replicate itself, but make mistakes that lead to evolution. Well, it's a simple molecule. It's not very complicated. And what we're going to see is that there are three main components that we're going to think about when we generate DNA. The first component is a sugar. It's a 5-carbon sugar called deoxyribose. And I'll spell out what this means. But then in addition to the deoxyribose, this 5-carbon sugar on the number 5 this 5-carbon sugar on the number 1 carbon will have a nitrogenous base. We know these as adenine, cytosine, thymine, and guanine. But in addition to the nitrogenous base in the number 1 carbon, the number 5 carbon can carry a triphosphate. And this will be, as you'll see, a relative of the adenosine triphosphate we talked about the other day. We'll obey many of the same rules, and we'll use the energy in this triphosphate bond now to be able to form phosphodiester linkages that will make a great long deoxyribose polymer. We're going to covalently link one deoxyribose to the other to make this great long strand of DNA, and we're going to link those sugars together by these phosphodiester linkages. But that's not enough to be a DNA molecule. To be a proper DNA molecule, one also needs to have base pairing. Now, where the polymers are dependent upon covalent bonding, the two polymeric strands of DNA are held together by base pairing. So this is going to be effectively an electromagnetic interaction because of the base pairing due to hydrogen bonds. So we're going to reintroduce hydrogen bonds here in a slightly different way. And that hydrogen bonding, again, is going to be based on charge, and it's going to hold the two strands together. But not only does it hold them together, but it also makes them more or less malleable. That is, you can separate them and bring them back together again. You have enzymes that allow this molecule to be plastic in that way. The other characteristic that we'll see here, because of the layout of the structure of the deoxyribose, because of the orientation of the phosphates, we end up having to produce a double strand that's said to be antiparallel. And there's a kind of nomenclature we'll walk our way through, so that one strand will be labeled 3' to 5', and the other will be labeled 5' to 3'. And this nomenclature is dependent upon the numbering of the 5 carbons on the deoxyribose sugar. But the molecule can't just replicate itself. In biology, it requires the addition of other and the assistance of other enzymes. The first enzyme that we'll talk about is one called a helicase. And you may recognize here the importance of this structure of this molecule and how it implies the structure of DNA. One of the hallmarks of DNA is it's thrown into this helix, two strands held together by hydrogen bonds thrown into a helix. That helical nature protects the inside of the hydrogen bonding. So in order to get replication of the two strands, what we have to do is first unwind the two strands, and that's the job of this enzyme, the helicase. It's not too much of a stretch to think of this as a kind of wedge being driven between the hydrogen bonds and separating those hydrogen bonds mechanically by the helicase. But again, there's this other molecule that's now a proper protein. It's a proper protein, it's a proper enzyme. And this enzyme, DNA polymerase, which was discovered in the 1950s by a guy named Arthur Kornberg, who really worked out all the details for this and got a Nobel Prize. This, in fact, this DNA polymerase is an enzyme that forms phosphodiester bonds. So we'll try then to link the idea of these phosphodiester bonds, these covalent bonds between adjacent deoxyribose molecules. We'll try to link this idea to the generation of strand and how that's done enzymatically. But there are a couple of ideas that will fall out of this. First is, when we follow this through, and it took a long time to sort this out, that it's something known as semi-conservative replication. What semi-conservative means is half of the new molecule of DNA is conserved and the other is new. The de novo strand, we'll see how that's put in, is made brand new, but it's made brand new because of base pairing with the old template strand. It's semi-conservative because each new molecule of DNA contains a template strand and a newly synthesized strand of DNA. But there are some hang-ups with this that I want to tell you about. There is this idea that the DNA polymerase always reads only in one direction. There's a directionality to the strands, and we have a DNA polymerase that reads 3' to 5', but we'll see that the two strands are organized in an antiparallel fashion. This brings up some problems for this enzyme, and we have to fix those problems and solve these problems in order to generate two new strands in this semi-conservative fashion. And once we get to here, then we'll go and begin talking about the cousin molecule, the other nucleic acid, ribonucleic acid.